Hello, my name is Josh Mahlios. I'm a Senior Marketing Product Manager at Miltenyi Biotec, and today's webinar will be on the automated cell separation for drug discovery assays. Today's agenda will be as follows. First, we're going to talk a little bit about the background of Miltenyi Biotec and how we are pioneers in cellular therapy. Next, we'll talk about the isolation of primary cells, moving beyond density gradient centrifugation. Next, we will talk about the automated isolation of primary cells. This is for reproducibility and scalability. And then finally, we'll talk about drug discovery assays and some example applications. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi, and the basis of max cell isolation is column technology developed as a part of his diploma thesis, and this was patented in 1990. Already in the beginning stages, Stefan Miltenyi had a pioneering spirit and saw the potential for MAX technology to become a revolutionizer in the field of gene therapy. The CliniMax CD34 reagent system is a method for selecting hematopoietic stem cells from donor apheresis while passively depleting T cells that can cause graft versus host disease. The development went on for over 20 years, including setting up a GMP manufacturing site in Teterow, Germany. And finally, the CD34 isolation using the CliniMax together with the CliniMax CD34 reagent, which received FDA approval in 2014. What started out as one man's diploma thesis has since grown into a global company of more than 2,500 employees worldwide and over 20,000 scientific publications citing MAX technology. Annually, more than 2,500 patients receive cell products manufactured with Miltenyi Biotec technologies. The phrases empowering discovery and advancing therapy get to the core of our company's mission, which is to improve scientific understanding and medical progress by providing solutions that advance biomedical research and cellular therapies. And we achieve this mission by our ongoing commitment of bringing newer and smarter technologies to the scientists and clinicians who are paving the way to longer and healthier lives. MAX technology is based on three basic principles. First, you have the magnetic labeling, where an antibody conjugated to a tiny paramagnetic microbead binds to a receptor or protein on the cell surface. There are different strategies for this step, which will be covered in more detail later in the presentation. Next, we have magnetic separation, the process where cells are passed through a column that is positioned within a magnet. The cells labeled with the microbead are retained within the column, whereas everything else passes through in a gentle way. The flow-through is called the negative fraction, containing the cells without the ligand or antibody that was used. The elution step. The column is removed from the magnet, enabling the paramagnetically labeled cells, meaning that the microbeads lose their magneticism outside of the separator, allowing them to pass freely through the column. This can be achieved through a quick manual step using a plunger to gently elute the cells, or via an equally gentle automated process using a vacuum to pull the cells out by increasing the flow speed of the buffer in the system. These methods are all gentle on the cells and separate them into negative and positive fractions with exceptional purity and high viability. MAX technology is therefore based on three key components: the MAX microbead, the MAX column, and the MAX magnetic separator. There are several versions of each available, and depending on your starting material, cell type of interest, and whether you're using a manual or automated system. Since the column is the heart of MAX technology, let's take a quick look at the benefits and why it is clearly the superior choice when compared to column-free methods. The magnetic separators in column-based and column-free systems have a comparable magnetic force. The difference between the systems is a column, filled with little iron spheres that pass the magnetic force over onto the next one over a very small distance, amplifying the magnetic force by 10,000-fold. The resulting benefit is that minimal labeling of the cells with microbeads is sufficient to retain the target cells within the column while in the separator. The iron spheres have a cell-friendly coating that allows cells to run freely through the column and can be washed thoroughly to remove cell debris and contaminants. All of this contributes to the fact that MAX columns allow for cell separation with minimal effects on cells. Combined, these advantages make MAX columns the best choice for cell separation for drug discovery applications. Before we get into the specifics of primary cell isolation, let's consider the use of primary cells and cell lines in the drug discovery process. Primary cells are sourced directly from human or animal tissues, whereas cell lines, on the other hand, are generated by immortalizing primary cells with vectors and cultivating them over many passages. The cell cultures that result differ vastly regarding the tissue of origin, cellular characteristics, and actual relevance to predict in vivo biology. Starting with primary cells, they have a relatively limited supply as blood donations are needed, and there's quite a bit of variability when it comes to the quality and consistency. These cells have a limited proliferation in culture, as well as a limited lifespan. You can see high donor variability, and depending on what you want, this can be either a positive or negative characteristic necessary for immunogenicity and biomarker discovery, whereas it's not wanted in screening or banking for optimization of molecules. These cells have a relatively low mutation rate, and this preserves in vivo characteristics, giving you no surprising modifications. It has the same genetic makeup as the original tissue. These cells have a more representative biological phenotype and complexity. The physiological and clinical relevant phenotypes that these cells offer are also a benefit. These cells are pre-characterized and are ready to use. And in comparing this to cell lines, there are a lot of differences. These cells are virtually unlimited in supply. They're easy to culture almost indefinitely. There's no donor variation. However, many cell lines are often misidentified and also can be contaminated with other cells. This gives you the danger of false positives. These cells have a relatively high mutation rate, and cell lines often mutate so that their genetic and phenotypic characteristics are no longer representative of those from the original donor cells, either through the immortalization process or afterwards. They have relatively low relevance in vivo and are often used on research that's focused on a very specific target. And lastly, the authentication that is needed in these cells is a major concern. Misidentified and cross-contaminated cell lines is a major issue. In a recent study of over 500 leukemia and lymphoma cell lines, 15% of those cell lines were misidentified. Most primary cells come from human-derived blood products, such as leukopaks, Buffy Coats, or whole blood. In the United States and Canada, Miltenyi Biotec is now offering leukopaks, bone marrow, and whole blood. If you have any interest in this, please visit our website or come to the booth for additional information. Tissues, on the other hand, can be of murine or human origin and can be either healthy, such as spleen or lymph nodes, or diseased tissue, such as tumors. These include patient-derived xenografts in mice, which are also often used in the drug discovery process. This sample preparation step is mostly density gradient centrifugation to get PBMCs, or in the case of tissue, dissociation of the tissue, for example with the Gentlemax dissociator, to get a single cell suspension. After this, you can proceed with the cell separation. Isolation strategies can vary depending on your project, and we have a wide selection of different options to suit your needs. Would you rather go for a fast and easy positive isolation, or do you need all of your epitopes free for a second sort, requiring an untouched isolation where you label everything else but the population of interest? Or do you have multiple samples and would prefer to separate them in parallel rather than sequentially? Are you freezing aliquots of your PBMCs or isolated cells directly? All of these factors are important when deciding on which cell separation strategy is right for you. Once you have your population of interest, you can move on to the downstream applications. Whether it's an additional separation step for subpopulations, for example, Tregs, or flow cytometry for immunophenotyping, we can recommend kits that will make these steps easier. Are you culturing your cells for in vitro assays or running genomics and proteomics applications? Whether you are interested in maximum recovery for cell culture assays or high purity for reliable genomics or proteomics studies, we can help you with the strategy to get there. We will now focus on PBMC isolation for drug discovery applications such as immunophenotyping and in vivo functional assays. To recap, traditional approaches go from blood products to PBMCs to target cells in 2 steps. In this process, you have to go from a blood sample to PBMCs via density gradient centrifugation. I'm sure that many of you unfortunately know this process quite well. There are a number of steps required to isolate PBMCs via density gradient centrifugation. First, you have to carefully layer your blood sample over the gradient, then centrifuge the sample for 20 to 30 minutes with no break, which is one of the most infuriating aspects of this process, followed by a highly variable PBMC recovery process and wash, and for subsequent target cell isolation, an accurate cell count. This process takes more than 2 hours and can easily eat up most of your morning depending on the number of samples that you have to process. StraightFrom MicroBeads are the hassle-free alternative to density gradient centrifugation. Our StraightFrom product line allows for the isolation of cells directly from blood products without density gradients, without cell counting, and in only a few simple steps. With minimal hands-on time and user variation, our StraightFrom products offer the fastest available protocol for target cell isolation from Buffy Coats and Leukopacs. The protocol is really simple. You transfer the Buffy Coat, Leukopac, or LRSC into a suitable container and fill it up to 80 mL if the volumes are less than that. This example is for a Buffy Coat, but it's similar if you're working with an LRSC or Leukopac. Add the microbeads and incubate in the fridge, after which you can separate the cells using the Multimax 24. We'll get to the instruments in a little bit. You can go from an entire Buffy Coat to target cells in as little as 25 minutes, or from half a Leukopac in 40 minutes. Finally, you have another option to density gradient centrifugation. Miltenyi Biotec is excited to introduce our StraightFrom MicroBeads for the direct isolation of target cells from blood products, and we've just launched our new products for Buffy Coats, Leukopacs, and LRSCs. To summarize thus far, we've now learned about primary cells, what we need to consider when planning a project to isolate cells, the steps to isolate them from tissue or blood products, as well as the shortcuts we can take. Everything we've talked about aims at generating assay results that are biologically relevant, reproducible, and as efficient as possible, saving hands-on time and unnecessary repetitive steps. However, the reagents we work with are only half of the solution. Truly efficient protocols with reproducible results require automation. So I'd like to take a few moments to discuss automated solutions before going on to discuss drug discovery assays. So what is the benefit of having automation? Automation reduces hands-on time and frees you to work on more other important tasks that cannot be automated. It minimizes variation between different users in the lab, or even good and bad days of an individual scientist. And of course, it minimizes costly human errors. With standardized protocols and minimal user variation, you can be sure that the effects that you're seeing are due to your assay rather than artifacts. In short, automation reduces redundant experiments and allows you to focus your time where it matters. Miltenyi Biotec has developed various automated solutions for the isolation of target cells for various drug development assays. We'll start with the AutoMax Pro separator, the easy-to-use benchtop instrument for every lab. With fully automated sequential separation of 1 to 6 samples, the AutoMax Pro provides easy access to automation without the high investment costs or lots of training. You can isolate cells from different starting materials in one run, press the start button, and the instrument will label and separate the cells for you. The columns are reusable, so maintenance is easy, and there are preset programs for high recovery and high purity depending on your downstream applications. If you have more than 6 samples, or large samples such as Buffy Coats, you might want to consider parallel cell isolation. The Multimax family is perfect for this. 24 columns allow you to process 1 to 24 samples in one run, or split your large sample between multiple columns. This is achieved by using the LS, LD, or whole blood columns, or the multi-24 block for easy handling of 24 samples or an entire leukopac. The Multimax Cell 24 is a semi-automated instrument and a good fit for many labs that value easy handling but don't mind the manual pipetting. However, if you want full automation and maximum throughput, you will love the Multimax X, which integrates a Multimax 24 into a liquid handler. Launched in 2017, this is the instrument for the demanding high-throughput cell processing lab. Samples are labeled and isolated automatically, and you can go from an entire Buffy Coat to target cells in 30 minutes, or from a leukopac in 60 minutes. All programs are tailored to meet your needs, and we won't leave your lab until you are satisfied with the protocol and the results. In summary, the AutoMax Pro is great for small samples and low throughput with full automation, for example when processing PBMC aliquots, mouse samples, or patient-derived xenografts. Because the AutoMax can process whole blood without Ficoll, it's also great for patient samples or small donor samples. On the other hand, the Multi-Max family are the high-throughput variants for fast parallel processing, for example of small human or mouse samples, but also for large blood products such as Buffy Coats and leukopaks. We've now learned about the steps of primary cell isolation and how to ensure maximum efficiency and reproducibility by selecting the best reagents and instruments to match your workflow. At this point, I'd like to take a chance to go through some of the application data from the assays frequently run in the drug discovery process at different stages. This includes phenotypic screening for target discovery, animal model development, and toxicity or immunogenicity. We'll start with phenotypic or high-content screening. Phenotypic screening can be used for biologic and small molecule development. The example here is small molecules, where we looked at the binding of MCP1 to CCR2 on the surface of monocytes using high-content screening in the development of an anti-inflammatory drug, a CCR2 inhibitor. CCR2 is expressed on monocytes and is the binding site for MCP1, a chemokine that attracts and activates monocytes. In the top row, you will see the blue monocytes on the left and the red MCP1 in the middle, and the overlay shows that the cells in the MCP1 are co-localized. In the middle row, unlabeled MCP1 was added in excess to block all monocyte binding sites, and as you can see, no stained MCP1 can be detected. The same is seen when the CCR2 inhibitor, which is the potential drug, is added, and there's no MCP1 signal since there are no free binding sites. Additional information can then be acquired on a single cell level, for example, the shape and size of the cytoplasm, as well as dynamic information about the nucleus. Another example of drug discovery assays that utilize cell separation is the development of mouse models. This example is the humanization of a mouse. First, you would irradiate the mouse, then transplant human CD34 positive hematopoietic stem cells isolated on the AutoMax Pro. After infusing these cells into the irradiated mouse allows you to grow a humanized immune system. Of course, you can then monitor the success of this humanization by isolating the human immune cell subpopulations after infusion. In the later stages of the drug development process, you will be needing lots of primary cells for the mechanism of action profiling of your drug candidates to understand the exact mechanisms by which the drug influences the immune system, which concentrations are needed, and in which combinations. In the example of a mixed lymphocyte reaction, you will isolate monocytes from one donor and T cells from another donor. You will differentiate the monocytes into dendritic cells and combine them with the T cells from the second donor. This combination will lead to the activation of the T cells, and by adding various checkpoint inhibitors, these molecules will unleash the brakes and enhance the reaction. The T cell activation can be tracked, for example, by interferon gamma production, as we will show on the next slide. The T cells and controls by themselves show low levels of T cell activation. However, when you add a PD-1 antibody or a PD-L1 antibody, you can show significantly increased interferon gamma production in all three donor pairs as a sign of the drug candidate enhancing the immune reaction. Another method of MOA profiling is the Treg suppression assay. Here you isolate CD4 cells and Treg cells. You simulate the CD4 cells and then combine them in co-culture. During the co-culture, you can track the inhibition of the T cell activation by the Tregs, and by adding the drug compound of interest, you can detect compounds that can suppress the Treg inhibitory effect, as you can see on this graph on the right. The same method can also be used for MDSC suppression assays, which are based on the same principle. As an example of a killing assay, in the T cell dependent cellular cytotoxicity assay, you isolate pan T cells and culture them with EGFR positive cancer cells. In this case, CD3 EGFR bispecific antibodies bring these T cells and cancer cells together for more efficient killing, which can be monitored in an ATP-based readout, for example, a cell titer glow luminescence, as in this example. Cell isolation is also essential in CAR T research. You can monitor the transduction, activation, and killing, as well as manipulate the effector to target cell ratios. For example, here you isolate T cells, transduce them, for example, with a prostate cancer-specific antigen, PSMA, and culture the cells culture the transduced CAR Ts with the two cell lines, one antigen positive and one antigen negative. A TDCC assay might follow to monitor the killing, as we just discussed. The output looks like this, where we have our PSMA positive cell line in black and the PSMA negative cell line in gray. In the first test on the right, you test for transduction efficiency with a CD19 CAR as a positive control. Comparing the two different CAR constructs on the top right, you see that the PSMA positive CAR T constructs are specifically activated using CD25 as an activation marker when in contact with an antigen positive cell cancer line. The lysis is equally specific, and this antigen-specific reaction is exactly what you want to see. Moving on from small molecules to biologics, we also have one example from predictive immunogenicity. In the MHC-associated peptide proteomics assay, you isolate the monocytes and differentiate them into immature dendritic cells. You then add the biologic drug candidate of interest. Afterwards, you can then precipitate the bound fragments to isolate the antibody fragments that were presented on the DCs and analyze the peptides by mass spectrometry to see which ones are presented and might activate the T cells. The mass spectrometer analysis of a therapeutic antibody with the heavy and light chains might look like this. You list all of your donors and map the peptides that are presented in a heat map for your specific antibody. This heat map can then be used to modify the antibody to de-immunize it and make it safer regarding immunogenicity. The next step of predictive immunogenicity is the DC T cell assay. Here you isolate monocytes that act as antigen-presenting cells. You co-culture them with T cells. And you add a candidate antibody or a mix of peptides from your fragmented antibody and see if CD4 positive T cells proliferate and produce IL-2, indicating that an immunological reaction to this peptide is occurring. This can be tracked by using flow cytometry, measuring the CD4 proliferation and IL-2 production using the cytokine release assay. On the right, you see the correlation of an in vitro versus clinical data that show that if a drug shows immunogenic genetic effects in the in vitro studies, it is highly likely to cause problems in the patient as well. Much of what we have talked about today revolves around the drug discovery process that is ultimately aimed at producing drugs for patients. To this end, we have a translational approach from benchtop to bedside that is reflected in all of our products. We provide the same products in research grade as well as GMP and clinical quality to ensure smooth transition into the clinic. We provide the entire workflow for products for cell-based work, from tissue dissociation and homogenization to cell isolation and culture, including activation and expansion, as well as cell analysis. So to summarize, primary cells are a valuable model for biologically relevant assay results. Reliable results depend on the right cell isolation strategies, and we've learned that you can reduce human error and improve efficiency with automated solutions in every stage of drug discovery. With this, I invite you to come and visit the virtual booth to learn more about our offerings. I thank you for your time, and please let me know if you have any questions. Thank you.